Hello friends, welcome to Tricks and Learn. So today is the first live is going on and it is the MCQ discussion from Biotechnology Principles and Processes. So it is an important chapter for both term to board exam as well as NEET syllabus. So today we are going to see the NEET previous year questions from Biotechnology Principles and Processes. So we already completed the chapters of human health and disease and microbes in human welfare. If you not yet seen, the video link is given in the description box, so you can also able to see in that link also. So, and another one thing, if you not yet subscribed to our channel, please the subscribe button and press the bell icon. Then only you will get the instant notification of my upcoming videos. So in this chapter, totally there are 65 questions. So the whole 65 questions we are going to see in the same video. So let's start. So first, 1 minute. So first question, the DNA fragments separated on agarose gel. They are given here is agarose gel. So it can be visualized after staining with which of the following stains are used here? Acetocarmine, aniline blue, ethidium bromide, and bromophenol blue. So here they are using ethidium bromide because ethidium bromide, the explanation given here is the separated DNA fragments can be seen only after staining them with a compound known as ethidium bromide. So what is the color it is giving? So it is giving the color of orange colored band. So it is giving a color, orange colored band is given. Ethidium bromide is the correct answer. Then second question. DNA fragments are, it is very easiest question. DNA fragments are negatively charged and negatively charged DNA fragments are collided with positively charged histone proteins. So it is a clue. So DNA fragments are negatively charged. So third question, the gene whose expression helps to identify a transformed cell is known as, the gene expression helps to identify the transformed cell. So which of the following gene expression is helps to identify the transform cell is selectable marker is the correct answer. So I think we already studied. So there are some genes, they are called selectable markers. They help in selecting those host cells which contains the vectors. What is vectors? Vectors are the transformant. So they are the transformant and eliminate the non-transformant. So because of this reason, option D is the correct answer, selectable marker. Fourth question, what is the criterion for DNA fragments movement on agarose gel during gel electrophoresis? So this question also we already saw, I think. So we know that the smallest fragment will cross the more area because we know it is agarose gel pad. So here we are going to use our sample and it is connected, and here completely we are going to use agarose, a gel. It is a water form, jelly form. So we are going to put the agarose gel and we are going to get the samples here. So we are going to put the samples here. So this samples contains genes. They are called selectable markers. So in principles of inheritance chapter and also molecular base of inheritance, in this chapters we studied about this gel electrophoresis, I think. And here when it is connected to positive and negative, the low density genes or DNA are move faster. The low density are move faster and the high density are only settled here. So the lowest density are move faster and highest density are settled down. So because of this reason, for this question option A is the correct answer. So what is option A? It is the smaller fragments. The smaller fragment, the smaller the fragment size, the further it moves. So it is the correct answer. Then fifth question, the process of separation of purification of expressed protein before marketing is called. So the process of Separation of purification of expressed protein before process is called downstream processing. Very very important and very very easy also to remember. So downstream process, we know that what is downstream process. So the process includes separation and purification of product. Separation and purification, so this process is called downstream process, and it is happened in downstream process only. So next question. Stirred tank bioreactors have been designed for. Stirred tank bioreactor. What is stirred tank bioreactors? So it has some stirrer. What is the stirrer used for? Is ensure the availability of oxygen throughout the process. So it ensures how many oxygen is there. Like that, it ensures the availability of oxygen throughout the process. So it is the use of stirred tank bioreactors. Seventh question. A foreign DNA and plasmid cut by the same restriction endonuclease can be joined to form a recombinant plasmid using. So it is very easy question. It is already be studied in biomolecules chapter as well as microbes in it is from principles of inheritance and molecular basis of inheritance. So it is DNA ligase. So what is DNA ligase? So for some DNA, it is not connected, it is not joined. So DNA ligase is a type of enzyme, so which attach here, and it makes the bond between these two. It makes the bond between these two, and it be able to connect this. So this is DNA ligase. So to join the recombinant, we are using DNA ligase here. Then eighth question. Which of the following is not a component of downstream process? We already saw what are the components of downstream process. So downstream process contains separation and purification. So here for eighth question, separation is also a downstream question, purification also downstream process, and preservation is also comes under purification, because the purity of a compound is separated. It is distinguished while purified. Preserving also very important, right? So because of this reason, so option D is the correct answer, because expression is not a final product, or it is not a component of downstream processing. Ninth question. Which of the following restriction enzymes produces blunt end? So which of the following will cause blunt end? Means we are going to make a discussion here. So eco or B, we know that eco or B. So it is a type 2 restriction endonuclease. It is isolated from the bacteria. We know that it is E. coli. And here it creates blunt end. This only creates blunt end, but SAL1, X-Hall and HIND3, these are product sticky ends. So I think you know about sticky and blunt end. So here for this question, option B is the correct answer, because eco or B produces blunt end and other three produces sticky end. Then 10th question. Which of the following is not a feature of plasmid? So not a feature of plasmid is option B, because it is single stranded. It is not a feature of plasmid. So feature of plasmid is it is transfer something from one to one, and it is independent of replication, and it has structural structure. We already know plasmids have a structural structure, circular structure. So here plasmid, it may be single stranded, double stranded or anything. So it is not a future, is single stranded is the correct answer. 11th question. The Taq polymerase enzyme is obtained from. The Taq polymerase enzyme is obtained from. Very easy question. The name itself it has. So it is Thermus aquaticus. Thermus aquaticus. So it is a type of thermophilic bacteria, and which can be live in high temperatures also. So it is an enzyme produced from Thermus aquaticus is Taq polymerase. Then 12th question. Which of the following is a restriction endonuclease? So it is very easy. HIND2 is a restriction endonuclease. So what is restriction endonuclease? They are molecular scissors. They are used to cut a plasmid DNA or cut a part of DNA to make it as a recombinant DNA. So restriction endonucleases or molecular scissors. They are also called molecular scissors. So here DNA is an enzyme, RNA is an enzyme, Protease is an enzyme, but it is not a restriction endonuclease. Hint 2 is a restriction endonuclease. 13th question. Which of the following is not required for any of the techniques of DNA fingerprinting available at present? So, restriction enzymes, DNA hybridization, polymerase chain reactions, and zinc finger analysis. So, which of the following is not required? Because these three we know restriction enzyme are used to DNA fingerprinting, and DNA we want to hybrid because one DNA we want to make it as a more DNA. This process is called polymerization, and this polymerization can be done by polymerase chain reaction. So this A, B, C are the required process, and zinc finger analysis is not required for this process. So option D is the correct answer for 13th question. 14th question. The DNA molecule to which the gene of interest is integrated for cloning is called. The gene of interest introduced for cloning is called. Okay, so we are going to see the options. Template, carrier, transformer, and vector. So very easy. I think you catch the answer. It is vector. So because what is vector? Vector is a DNA molecule that carries a foreign DNA sequence and replicate inside the host, not outside the host, inside the host cell. Okay, this vector DNA and a foreign DNA carrying gene of interest. So it is cut by the same endonuclease enzyme. So while cutting with the endonuclease enzyme, this two produces sticky ends. So it is very easy to note that it is produce sticky ends, not blunt ends. It produce sticky ends. So it produce sticky end means it can be the enzyme of SAL1, XHOL1, hint 3 because we already saw in the question number 9. So in 14th question, the correct answer is vector. Okay. 15th question. The cutting of DNA at specific locations became possible with the discovery of which one will causes possible cutting of DNA is the 15th question. So it is restriction enzymes. So everybody know restriction enzymes or restriction endonucleases or the molecular scissors. They cut the DNA at specific locations to make it as a recombinant DNA. So 1 minute break. Okay, 16th question. So 16th question, the analysis of chromosomal DNA using the southern hybridization. So we want to note here is, so here we want to note is southern hybridization. So in southern hybridization, the process which involves is PCR, not involved is PCR. Because what is PCR? PCR is a polymerase chain reaction, and in PCR we do the amplification of the DNAs. So in this lesson, so this PCR technique is mostly involved for amplification. For example, we contains only one DNA means in this amplification process we are going to get more similar copies of DNA. So this process is called an amplification, and it is carried out by PCR, polymerase chain reactions, PCR. Then 17th question, which vector can clone only a small fragment of DNA? So the vector which only clone a small fragment of DNA is called plasmids. So it's directly from NCRT. So the vector which is clone a small fragment of DNA is called plasmid DNA. 18th question, commonly used vectors for human genome sequencing. It is commonly used is option B, BAC and YAC, bacterial artificial chromosome. So these two are the commonly used vectors for DNA sequencing, that is human genome sequencing. So it is directly from NCRT. 19th question, the colonies of recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria. So because of, for 19th question, the answer is option C, because